Thank you for coming to my very first TIS webinar. I'm very excited to talk to you today about ensuring research resiliency with efficient mouse colony management. To maximize colony efficiency, it is really important to tailor your colony to your experimental goals. This requires understanding several key colony management principles. We will talk first about mouse reproduction and the reproductive milestones and a mouse's reproductive timeline. Then we will discuss breeding performance, including genetic and environmental factors that can influence it. Third, we'll talk about data collection and good record keeping. And then finally, we will discuss the role of genotyping in maintaining your colony. Good colony management can have strong effects on many parts of your research. By implementing thoughtful colony management practices, you can support the 3 Rs, improve experimental reducibility, and reduce costs. Before we move into the first topic in our speech, I would like to touch on the 3 Rs and their importance in colony management. If you are unfamiliar with the 3 Rs, they were defined by the National Center for the Replacement, Refinement, and Reduction of Animals in Research. The 3 Rs are important in promoting research resiliency. They encourage high-quality research by in-depth examination of model selection, an increase in efficiency for colony management, and help maintain the ethical use of laboratory animals. The first R stands for replacement, which discusses methods that avoid or replace the use of animals by using things such as mathematical or computer models and established animal cell lines. The second is reduction, methods that minimize the number of animals used per experiment or study by enabling researchers to obtain comparable levels of information from fewer animals or to obtain more information from the same number of animals, thereby avoiding further animal use. Practices that fall within this reduction category include improved experimental design and statistical analysis, sharing data and resources between research groups and organizations, and the use of technologies such as imaging to enable longitudinal studies of the same animals. In the context of today's topic, implementing good colony practices can help reduce things like reducing mice with unwanted genotypes, reducing costs associated with maintaining your colony by choosing a colony size that fits your research, and by thoughtful and careful breeding practices. Choosing proper controls can also reduce the resources required for statistical analysis, as well as reduce your headaches when it comes time to publish. The third R is refinement, which means implementing methods that minimize pain and suffering, distress, or lasting harm that may be experienced by the animals. Refinement applies to all aspects of animal use, including housing and husbandry, to the specific procedures performed on them. Refinement includes using appropriate anesthetics and analgesics, as well as avoiding and minimizing stress by training animals to communicate or to cooperate with procedures such as blood sampling, and by providing animals with appropriate housing that allows the expression of their species-specific behaviors, such as nesting mice. When maintaining mouse colonies, it is important to remember that the most basic component of a mouse colony is the mouse themselves and their ability to reproduce. To manage your colony effectively, you need to understand the strain you are working with and the reproductive capabilities of those mice. Researching your mice thoroughly before beginning a colony is going to help you save on costs and time by promoting more successful breeding once the mice are in your facility. Understanding factors that can affect breeding performance, such as genetic background, the environment, and mutation or transgene effects, is an important part of efficient colony management planning. Let's go into depth about some more of the factors that are important to know. Strain differences can impact your breeding efficiency. Being familiar with some of the common reproductive characteristics of your specific strain will help with efficient colony management and maintenance. Things such as gestation. Gestation is generally consistent between individuals of any given inbred strain, but you will see some variation between strains. For example, C57 Black 6 is close to about 18.5 days. However, Bald C tends to exhibit closer to 20 days, and the AJ will show a 21 day. Gestation is also affected by litter size. Larger litters tend to be born earlier, and gestation is often longer in older females. Weaning age depends upon the size and development of the pups. At jacks, most strains wean at 3 weeks. Some strains, however, benefit from delayed weaning if the pups tend to be smaller. For example, cast EIJ, they tend to have very, very small pups, and we have moved to a 4-week wean for them. 21 days is the earliest that we will ship any of our mice without a lactating female. Sexual maturity, typically 5 to 6 weeks in females and 6 to 8 weeks in males. However, this can potentially vary by strain. Optimal breeding lifespan is approximately 8 months, but it will depend strongly on the strain. AKRJ develops leukemia, and breeders typically are replaced at about 5 months of age. The NOD LTJ females develop diabetes as early as 17 weeks of age, and breeders are retired at 18 weeks. Older inbred mice will breed, but the litter size and frequently typically declines after that time frame. Strain background can have effects on many factors that influence your colony management, such as birth defects. Certain birth defects occur more frequently in some strains than others. C57 Black 6 has more or a higher incidence than average of things like hydrocephalus and malocclusion. Total fecundity, the total reproductive capacity of the mice. Are they good or poor breeders? Wild-derived inbred strains, such as the CAROLI, the Caroli, or the cast EIJ we just mentioned, are known to be poor breeders and take some extra care. Hybrid mice tend to breed more vigorously than inbred. Litters tend to be larger, more frequent, and the mice may breed more successfully for a longer period of time. This increased breeding vigor of hybrids reflects the greater genetic heterogeneity compared to their inbred parents. Inbred mice tend to be bred to homozygosity in most of their genetic loci. This process means that mice more often become fixed for one or more recessive alleles that may negatively impact their breeding performance. Thus, inbred mice typically breed more poorly in comparison to their outbred parents. Crossing two inbred strains to make an F1 hybrid can reverse this phenomenon because the chances that two strains are homozygous for the same deleterious allele is very small, and the phenotypic effect of those recessive alleles can become masked by that wild type allele that is contributed by the other parent. Therefore, we tend to see an increased breeding vigor in hybrids. Behavior can influence the success of litters. Things such as poor mothering. C57 Black 6 females often cannibalize their first litters. Or you can see aggression. Although the exact cause of mouse aggression is unknown, taking steps like reducing stress or considering enrichment may help. However, some strains, such as SJLJ, are known to be more aggressive than average. Aside from genetic background effects, genetic engineering, such as the mutations for transgenes, can also affect breeding performance. Mutations are transgene effects that can lead to embryonic lethality or infertility, leading to no litters, small litters, or pre-wean mortality. For example, stock 006494, shown here, show a difficult in caring for their pups due to the disease onset. You may need to be aware of how a mutation or transgene can affect lactation. For example, mice that are homozygous for the DGAT1 knockout are void of milk production and require the use of lactating foster mothers. Some strains exhibit a shortened breeding lifespan due to certain mutations or transgenes. Example, mice that are homozygous for the TRP53 knockout develop lymphoma at about 4 to 5 months. Another example is that the strain that you see here. These mice display a progressive neurological phenotype that mimics the feature of Huntington's disease with an onset of approximately 18 weeks, shortening their breeding lifespan. The severity of these defects may be affected by the genetic background of the strain. Although the mice may breed fine on one background, they may not breed as well on another, so it's really important to know your strain background. While we mentioned the effect that genetic background can have on colony planning, it's also important to consider the effect of this background in conjunction to the strain's mutation and how that phenotype is integral for efficient colony management. Let's take a look at this interleukin 2 knockout mutation. On a mixed background, the average lifespan is between 4 and 9 weeks, and it develops progressive colitis. Due to the lifespan of these mice, maintaining a homozygous colony would be a challenge as the mice die prior to successful mating. Choosing a different breeding scheme, such as heterozygous by heterozygous, would allow you to obtain homozygous mice for an experimental cohort, additional heterozygous mice for colony maintenance, and wild-type mice for controls. When this strain is made congenic on C57 Black 6, the mortality increases to 10 to 25 weeks and still shows the development of progressive colitis. The increase in the lifespan may allow you a very limited window to set up homozygous by homozygous matings, but it would be important to consider how many more pairs or trios that would be required to maintain that colony, and depending on your research needs, that might not be an efficient use of your resources. Now, when this strain is made congenic on the BALB-C inbred background, the mortality is observed around 3 to 5 weeks with no progressive colitis, which could be due to the fact the mice probably don't live long enough for it to develop. Building an experimental cohort with this strain will take additional consideration due to the early mortality of the homozygous genotype. Breeding is highly influenced by environmental factors. Environmental stressors are one of the most common factors that we see influencing successful breeding. These things are typically the most challenging to deal with in your colony because you often have little control over them. Some strains will never be good breeders, but these are steps you can take to try and optimize productivity, hopefully saving you time and other resources. The commonly used temperature range for a mouse colony room is approximately 18 to 24 degrees centigrade. It may be helpful to verify temperature regulation for your room and also perform routine monitoring and maintenance of your rooms to ensure that they remain in an acceptable temperature range. The light cycle and intensity is very important. Mice are primarily nocturnal and they mate in the dark. Ovulation occurs in the middle of the dark cycle, and females are only receptive to mating for a very short window following that ovulation. Two frequently used cycles include a 14-hour on, 14-hour off, and a 12 on, 12-hour off. It is important that the dark cycle is not disrupted because this is critical for successful mating. Noise level can cause stress for mice. In our facility, we do our best to keep the noise level in the rooms below 65 decibels. It is really important to be aware of vibrations from the environment surrounding the room, such as construction in surrounding areas. A colleague of mine spoke of an incident where heavy construction going on about a mile away from their vivarium, and the deep ground vibrations from it did affect the breeding performance of their strains. Minimal handling, but especially near birth, such as delaying changing the cage until a few days after birth, is really important. When handling a mouse, a calm demeanor is best. Handle mice with gloves or forceps. Disinfect each between cages. And if bare hands are allowed, you can change your gloves between cages. Make sure you feel completely comfortable handling mice before you become their primary caregiver, so you don't have a direct impact on their breeding. Some strains react poorly to caretaker changes, and it may take them several months to adjust to a new handling regimen. Mice have an extremely well-developed olfactory sense and are very sensitive to odors, including perfumes or cigarette smoke. Here at the Jackson Laboratory, we do not allow anyone who handles the mice to wear perfume. Be cognizant not to spread scent from one cage to the next, especially if handling pregnant females. Nutrition is really important to monitor. Some strains show improved breeding resources with different dietary fat. BALB-C, J, showed improved performance here at the Jackson Laboratory with a 10% fat diet. Our Cath-EI-J breeders were historically maintained on a 4% diet, but showed improvement when they were changed to the 6% fat diet. We also noted that reducing variability in dietary factors can help stabilize breeding performance. It's important to pay attention to the health status of the colony room. If mice stop breeding, check for pathogens and consult with your facility's veterinary staff about potential causes. Knowing information about challenges you may find with a strain can also help with efficient colony management. Let's discuss a couple of examples, C57 Black 6J and SJLJ. Barbering in C57 Black 6J can be a challenge when maintaining a colony. Barbering appears to occur more frequently in co-housed post-wean females and involves whisker picking and alopecia. The frequency increases with age and with cage density. We performed a larger scale study to compare the two types of enrichment that mice seem to prefer, shelter and activity, to see if this had any impact on the hair loss in the C57 Black J mice. So we broke them up into 3 groups, and what we found was that shelters diminish hair loss better than the activity enrichment. So on the x-axis, you'll see that that's the age in weeks, and on the y-axis, you see the percentage of mice with hair loss. And when given the activity, which is shown in blue, you'll see that we actually saw an increase of mice with hair loss. With no enrichment, you can see it forms a bit of a curve, and that one's in black. And in red, you'll see when tunnels and domes were introduced, that we saw a much less occurrence of that barbering. Another example of a strain-specific challenge would be aggression in the SJLJ mice. This aggression can impact strain breeding performance by reducing pup survival and the number of litters, as well as increasing the time before the first litter is produced. This aggression can be age and cage density dependent. While the cause of mouse aggression, as I mentioned earlier, is not fully determined, removing stressors and considering enrichment may help. So that is what we did. A study was initiated to look at the effect that different enrichment types had on reducing these aggressive tendencies. So you can see the examples in this slide. In addition to the aspen chip bedding, the mice were provided with nestlets, aspen shavings, alpha twists, or Enviro Dry. We found that the alpha twist and Enviro Dry increased the productivity in the strain by increasing pup survival and the numbers of litters each female produced, as well as decreased the time before the first litter. On the left, the number of pups that survived to the weaning increased with the use of these 2 enrichments. The center graph shows the average number of litters per female, and the right represents the data for the days until the first litter, which you can see has decreased. The important takeaway is to remember that what works for this strain might not work well for another, and that this solution worked for our colony, and this may not be the best solution for your facility. Taking these factors into consideration will give you a better foundation on how to efficiently manage your colonies. Once you receive mice and start a colony, it is important to start collecting your own information on how the strains are breeding in your facility. Now that we've discussed factors that affect reproduction, let's discuss the importance of data collection and good record keeping. What type of data is important to collect, and how can it be used for good colony management? Good record keeping is essential for maintaining a colony efficiently. Not only does good data improve efficiency, it also enables earlier detection of problems and trends. If you have poor colony records, you may not notice that you're getting fewer litters from your breeder pairs until several months have passed. A pedigree book is one of the easiest and best ways to keep accurate colony records. Often this is considered time-consuming to establish, but it is critical for maximum colony efficiency. Pedigree books can be both a physical book or an Excel spreadsheet or both. It is important to collect and evaluate breeding statistics for your colony regularly. Look for trends in things like litter size, gestation, time before first litter, those kinds of things. You will likely want to record all the strain data, including proper nomenclature, the genetic background, and the history of the mice. For us, history means before they came to the Jackson laboratory. You also want to assign a unique animal number or ID for each mouse in each strain. This is especially helpful if you are using an electronic method of record keeping. You will also want to have detailed records on pedigree information. You want to have details on the dam and sire that have been mated, including their mated date. You want to keep track of the litters for each breeder, as well as birth dates and the number of pups born. This lets you know ultimately how productive your breeders are in general. You also need to be aware of details about your weaned pups. Just because you have 9 pups born, you may not have 9 pups to wean. Details about pre and post wean loss is important to keep track of. Other considerations include things like you may not need many females in your colony and may consider not weaning females. Or you're only keeping homozygous mice, so you are not weaning heterozygous mice. With that being said, you may also want to record the sex and genotypes of your weaned mice. It is helpful to keep track of the generation number. This is important in preventing potential genetic drift in your colonies and will help you decide when to consider a backcross to help maintain a consistent phenotype or when to consider a different control selection. Let's talk a bit more about generation numbers. Typically, N is used to track the number of backcrosses, and this reflects relative genetic contribution of the inbred strain to the current strain. F is used to track the number of brother and sister matings. This is important for pedigreed colonies. When looking for a strain on our website, you may also encounter the following notations as well. P indicates the generation which has been cryopreserved in the strain history. Plus, it indicates generation information prior to the arrival at JAX and after its arrival at JAX. A question mark would indicate that the generation number is unknown, and you'll see a couple of examples here. When gathering information about a strain's history, obtaining information like this might be helpful. JAX provides strain data sheets for each of our strains, and these data sheets are a quick way to obtain a wealth of this information. For record keeping in the mouse room, here are a few tips that might make things a bit easier. Anything that you can preprint and bring into your animal room will help make sure that the data recorded is accurate and thorough. It can be hard to remember everything, but cards or forms can prompt user to input all necessary data. Up until this point, we've discussed factors that influence mouse reproduction and the importance of colony planning and records. Now let's touch on some good husbandry practices that can help ensure your success. Good colony management practices. So choose the location of your cages carefully. Mistakes with nomenclature can be very costly and difficult to sort out. Keep the mice of the same strain together, as well as separating strains that might have similar nomenclature to prevent any potential errors, such as the 2 that you see in the slide here. Colored cage cards are also a helpful way to minimize handler error. We all have made mistakes when we are tired. Building in systems to help prevent these mistakes will save a lot of resources over time. You will want to maintain a pedigreed colony using filial mating scheme. With filial matings, recessive mutations tend to be identifiable sooner. Having a pedigree can help prevent deleterious alleles from spreading throughout your colony undetected. And we'll talk a little bit more about this in a second. To help mitigate this potential genetic drift, about every 10 generations, you should refresh your colony, either by backcrossing to the inbred background or F1 hybrid background, or importing breeders from the same strain from a trusted vendor. If you are maintaining good colony records and evaluating those records frequently, then even any deviations will become apparent. These records could shed some light on whether you've had any changes in the environment or any potential breeding errors. Remember that I mentioned the background of a strain can impact breeding performance. If you need to change the background of your mouse for whatever reason, you may also see changes in breeding performance and phenotype. So let's take a little bit closer of a look about the role of a pedigree. So we begin with initial founder animals, say 2 breeder pairs in this example, and these pairs are bred together to get 2 litters of mice. From those litters, we will establish mating between siblings of litter 1 and then separately for litter 2, establishing 2 different pedigreed lineages while never interbreeding the descendants from the original founders. We continue to breed brother-sister within a lineage. In this type of scheme, recessive mutations may arise and become fixed, and can sometimes result in a visible phenotype in the affected strain. In this scheme, recessive mutations, if an unwanted spontaneous mutant shows up, such as in Pedigree 2, then this pedigree can be discontinued, and Pedigree 1 can be used to establish a new pedigree and continue the colony. Because you have pedigreed lines, it is less likely that that mutation has spread through the entire colony. However, if you maintain this breeding scheme long enough, so about 20 generations, you can generate your own substrain. Preventing substrain development and mitigating the accumulation of spontaneous mutations by refreshing your colony after about 10 generations can help maintain experimental reproducibility. Even with following all our tips on maintaining a well-managed colony, it can still get quite expensive. To keep your costs down, consider the following. Sizing your colony for what you have going on now, not what you think you're going to need 2 years from now. Colony sizes can be increased or decreased with just a bit of planning if your needs change in the future. When you are planning your experiments, are there ways in which you can reduce the number of animals and still achieve your desired experimental results? Looking at similar published studies can give you a good idea of typical cohort sizes. However, could reducing this number still give you the statistical power that you need? Can you use both sexes? Can you use mice of a certain age range? You can consider ordering a cohort of mice for a specific experiment rather than committing the resources to building up your own colony. Vendors such as ourselves can frequently provide large cohorts of mice. Considering donation of unique strains to other repositories, when you can view this as immortality for your strain. So donated strains are cryopreserved, genotype verified, usually maintained with curated information, and are available in perpetuity. For strains that are in low use and you don't think you're likely to use in the next 6 to 12 months, consider cryopreserving your strain. Cryopreserving your strains can be a good way to cut down on colony maintenance costs, protect your strains from genetic drift, and has the added benefit of protecting your strain during things like disease outbreaks, natural disasters, equipment failure, breeding error, contamination, or genetic drift. One important factor in maintaining a colony that is frequently overlooked is the importance of genotyping your mice. Genotyping can help you set up your breeding with confidence and prevent costly mistakes down the road. For this section, I am making the assumption that everyone here is comfortable with the concept of Mendelian inheritance and its expected breeding ratios. Genotyping is important because it promotes accurate breeding and more reliable experimental cohorts, as well as prevents costly mistakes that decrease experimental reproducibility. When choosing a genotyping assay, it is important to choose an assay that will genotype for all the mutations or transgenes and can distinguish between all the genotypes that you might have. If you work with strains such as stock 029721, shown here, which carries multiple mutations, and there's a possibility that you might have multiple genotypes for each mutation, you will need to have an assay designed for each of those mutations. In this stock, the mice are compound heterozygotes for 2 mutations at the frataxin locus. One is floxed and one is a knockout. It may be helpful to genotype for both of these allele versions to verify the genotype of your mice. Frequently, in conditional ready strains, such as LOX-P flanked or FRIT flanked strains, once the recombination has occurred, your assay might not be able to distinguish the recombined allele. It is important to verify that your primers will distinguish the recombined allele. In the figure on the left, you can see an example of a LOX-P flanked allele. The green triangles signify the LOX-P sites. This example shows that exon 2 of this allele is floxed. If we take a bit closer of a look at this floxed allele, you can see where a typical set of primers shown in red for one of our assays may bind. After the pre-recombination, exon 2 and one of those LOX-P sites is no longer present, leaving only one LOX-P site and one primer binding site. If you are going to be genotyping multiple strains in a single plate, especially if the mice have similar mutations, such as multiple Cree strains, it is important to make sure that your assay is as specific to those different strains as possible to help ensure that you can trust your genotyping results. For example, if you are running a genotype assay that includes all of the 3 Cree mutations that are listed on the left, your temptation may be to run a generic Cree. However, choosing allele-specific Cree gives you the confidence that your mice type for the correct Cree, and you do not run into potential issues with Cree expression in unexpected areas due to a genotyping error. Many vendors provide genotyping assays for their strains. However, you can also frequently find primers used in the original publication or in other literature. If none are available, you may need to design your own assay. Please note that all assays need to be optimized for your facility, equipment, and reagents. Many assays will come with the conditions such as cycling times already included, but some do not, like ours at the Jackson Laboratory. Many of our conditions are influenced by the reagents that you are using during your PCR process. It is helpful to see the suggested conditions for your reagents and compare them to the conditions suggested by the assay. While they rarely differ substantially, a 10-second versus 30-second extension time can show very different results. In the example on the right, the top gel shows samples that were run for a longer extension time than the same samples run with a shorter extension time on the bottom. The top gel allows you to more easily and confidently score your genotypes. The extension time is where your Taq polymerase is adding the dNTPs to your template strands. If you do not give the Taq polymerase enough time to build these, then your amplification will be unsuccessful. Frequently, optimization can be done by running samples in gradients. By this, I mean running them with different concentrations and conditions to find the best amplification for your equipment and reagents. Optimization usually requires having mutant DNA available. Gradients can look like running a series of DNA samples, including a non-diluted and samples diluted to 1 to 10, 1 to 100, and 1 to 1,000 with water. If you know the concentration of each DNA solution, a 25 microliter PCR reaction typically requires about 5 nanograms of highly purified DNA or 40 to 50 nanograms of quick prep dirty DNA. In this example, different annealing temperatures were ran on the same samples, and you can see the presence of artifact or nonspecific bands are influenced by that annealing temperature. You can also try different extension times at each concentration to see where you get the best well-defined bands. It is important to run the following controls in each of your PCR reactions. Controls will provide you with invaluable insight when it comes to troubleshooting and will help you confidently interpret your genotyping results. For example, imagine that you have a breeding scheme in your facility that is hemizygous by wild type. If you run your assay with just a wild type control and see no mutant bands, it can be difficult to determine if the lack of mutant bands is from suboptimal amplification or true absence of the mutation in your sample. Having a positive control including in this sample will quickly tell you which one it is. A non-template control, usually water, is designed to look for contamination. Any bands that amplify here are likely also incorrectly appearing in your other samples. In this example gel, the scientist used a heterozygous control to control for both the wild type and mutant bands, and her non-template control is on the end. If one band is missing, the scientist can quickly assess using the control whether that primer set is amplifying adequately. And if the mutant primer did not amplify in her control heterozygous sample, she could quickly determine that her mutant primers may be causing this issue. Let's look at how detrimental a genotyping mistake can be for your colony. You have a hypothetical colony of transgenic mice that you are maintaining with a hemizygous wild type breeding scheme. You have purchased 2 mice and you set them up as a breeding pair. After they have offspring, you genotype the offspring and you include a non-template control and a wild type sample control in your samples. You find that you've produced 3 hemizygous offspring and 3 wild type offspring. You pair up the next breeding pair, and in time, you genotype these, including a non-template control and a wild type control. All 4 samples from these offspring amplify a perfect wild type band and no mutant band. You conclude that all 4 of these offspring from this pairing are wild type. You may think it's a tad odd, but Mendelian ratios are rarely perfect in each litter. You take one of these wild type mice and use them in an additional pairing. The rest you discard as additional mice are not needed. Additional wild type mice are not needed. You realize you have no hemizygous mice left, and you have to order another transgenic mouse to continue your breeding. After that mouse arrives, you set up a pair, then you genotype their offspring. You include the same controls, non-template and a wild type control. You then genotype their offspring. You include that non-template and wild type control. The pairing looks like it produced only wild type mice, and now you are concerned there is an error. You re-genotype all of the samples, and once again, you get a perfect wild type band and no mutant band. So you're probably asking yourself, did I get the wrong mouse? Unfortunately, unbeknownst to you, your mutant primers were left out a bit too long when you genotyped those F1 mice, and now do not amplify. This is what the actual genotype of the second litter was. When you selected the wild type from that colony, you actually selected this mouse, which is hemizygous for your transgene. But when you set up the next pair, you actually set up a hemizygous mating, and the offspring are a mix of homozygous, hemizygous, and wild type. If you caught the error here, you have discarded two valuable hemizygous transgenic mice from the previous litter, had to purchase additional mice, and if some of the previous misgenotype mice were used in an experiment, that data may now be compromised. All of that trouble can be solved by including a positive control in your PCR run. Controls are essential to good genotyping practices. Controls will provide you with valuable insight when it comes to troubleshooting and will help you confidently interpret your genotyping result. How do you get good positive samples? There are a few ways. Some vendors have control DNA available for purchase. You can use different strains that carry the same mutation if you know that the primers will amplify in both strains. And you can use the mice you order. It is always a good idea to genotype all mutant mice you receive. Not only will you feel confident about beginning your colony, but you can also use that DNA to optimize your assay and keep the DNA in aliquots for use as positive controls for future genotyping assays. Even if you maintain a colony as homozygous by homozygous, and it is extremely unlikely another genotype will be produced, genotyping your breeders will help ensure experimental reproducibility by eliminating potential breeding mistakes and providing you with confidence in your cohort. Some labs may decide to keep aliquots of DNA from their previous genotype mice for a certain amount of time. If you do discover an error, this can help you identify when the error occurred and which mice were affected, which may help you determine if you're still able to use some of your previously gathered data. It can also be used for retesting samples if there is a concern about the integrity of those results, and those samples can be used later as controls if they are needed. Today, I spoke about the components for effectively maintaining a mouse colony. Colony maintenance requires plenty of planning and oversight, but a carefully maintained colony can help ensure your research is consistent and reliable for years to come.